Okay, in today's class, I'm going to explain about the syllabus for first 20 to 25 minutes and later we'll continue with the leftover syllabus. Okay, so as I said before, totally 11 units. In that, I'm going to discuss this general concepts, non-core data, core data, and histology. Okay, in that, yesterday we have completely finished the general concepts. That is level of structural organization. Level of structural organization is nothing but they are going to deal with the structure, that is unicellular. Unicellular means it has a single cell, and that single cell is going to perform every other functions required by the organisms. And the organisms prefer to live colony, they prefer to live as a group. Okay, and they are also exhibiting multicellular forms. Multicellular form means they are made up of many cells, and each cell are destined to do several functions. And what about this prokaryotic and eukaryotic cells? This organism which has a very poorly developed nucleus is said to be as prokaryotic, and very well developed nucleus and very well bound membrane bound organs are said to be as eukaryotic cells. And the levels of organization of tissues, organs, and system. There are approximately 4 different grades of organization, that is cellular level of organization, tissue level of organization, organ level of organization, and organ system level of organization. In that, cellular level of organization is deals with an organism which is consist of only one cell, and that one cell or many cell, but that cell does not form any type as tissue. Each cell is destined to do different function. And what about this tissue level of organization is a group of cells joined together, and they are looking similar and they are going to do the similar function. Okay, and that is said to be as the tissue level of organization. For example, fungi. An organ level of organization is said to be as a group of tissue is going to give rise to an organ, and that particular organ is going to do certain function. Each organ is going to do certain function. For example, a kidney. In kidney, we are that is destined to do excretory function. For example, lungs. Lungs is destined to do a respiratory function. For example, plants. Plants is considered as an organ level of organization. An organ system level of organization means many organs comprises to form a single system, and that system, it consists of many organs like lungs, kidney. In that lungs, alveoli is considered as an air sacs, and that is where the exclusive exchange of air is going to happen. That is called as organ system level of organization. And as I said in the previous class, animals are said to be as the animals are upgraded to the organ system level of organization. In this, we have to understand why plant is said to be as the organ level of organization, and why animal is upgraded to the organ system level of organization, which is nothing but plant can satisfy their own needs by standing in their own place, that they doesn't need any type of locomotion or any type of locomotory organelle, so it is stop up to the organ level of organization. But in case of animals, it is heterotrophic. It needs to move from one place to another in search of food, nutrition, and whatever they need, they have to move from one place to another. So the animals is said to be as the organ system level of organization. Clear? Now, that's all about this level of structural organization. And the second one, acoelomata, pseudocoelomata, coelomata, proterostomia, and deuterostomia. What is coelom? In yesterday's class, I have said what is coelom. Can you please mute Mr. Ziomi? Coelom means a body cavity. One second. Coelo means body cavity. As I said in yesterday's class, there is three types of coelomates. That is acoelomate, absence of body cavity; pseudocoelomata, false body cavity; and coelomates, a true body cavity. And what are all the phylums which are placed under this acoelomata? What are all the phylums which is placed under the pseudocoelomata? What are all the phylums which is placed under the coelomata? That will be the questionable area. And one more thing is, there are two different types of coelomate organisms. That two types is schizocoelomate and the enterocoelomate. In case of schizocoelomate, what is the difference between these two means? The body cavity which is emerged from the by splitting the mesoderm is said to be as the schizocoelomate. In case of enterocoelomate, it is the body cavity will be emerged by the from the digestive system, that is said to be as the enterocoelomate, right? And Protostromia and deuterostromia is said to be the division. In that division, acoelomata, pseudocoelomata, coelomates are the subdivision. And the most important thing is which phylum is placed under which type of category. That will be the important area. And what is the concept of species and hierarchical taxa, binomial nomenclature, classical methods of taxonomy of animals? We should know why classification of organisms is important and why we want to study that. And one more thing is, in yesterday's class, I have explained the taxonomical classification of each organisms. For example, what is species, what is genus, what is family, class, order. That order of the hierarchy is very important. I have explained this hierarchical taxonomical classification with homo sapiens example, right? Okay, that's all about the general concepts. And when it comes to the non-core data, you have to keep in mind the important thing as the example plays a crucial role when it comes to the questions with respect to this non-core data, clear? And before we have to read this each topic given in the non-core data and core data, you should know what is the difference between this non-core data and core data, and what is the difference between the invertebrates and non-core dates. Okay, and what is the difference between the notochord and the vertebral column? You, before you should enter into each topics, each subtopics, you should know the basic difference of these two. If you know even the basic things, you can easily answer the question. Okay. Okay, when it comes to the non-core data, the first thing they have given is general characteristics and classification of invertebrates up to the class level. What they are and what they implies are, see, there are 9 different phylums: Porifera, Coelenterata, Helminthes. In Helminthes, there are 2 different phylums. One is Platyhelminthes and another one is Aschelminthes or Nemathelminthes. These 2 phylums are placed under the Helminth category, and another one is, next one is Annelida, Arthropoda, Mollusca, and Echinodermata. For all these phylums, you have to read the general characteristics and the classification. Also for Protozoa. Clear? After reading this general characteristics and classification of all the phylums, you have to read up to their class level. Okay, for example, see for example, take for an example Protozoa. You have to read the general characteristics of Protozoa and classification. This is the classification, this is the characteristics. This is the general characters, this is the classification. You have to read these general characters and classification for all the phylums, and after reading that you have to read what are all the subphylums and what are all the class and superclass and what are all the class. We have stop up to this, but we have a family, order, genus, species, that and all not necessary, but up to the class level we have to read all the classification. And the most important thing is example is an important thing. For example, the best example for Sporozoa is Plasmodium. Like this, you have to read for all the phylum. Okay, right. After reading the general characters and classification, you have to, then only you have to touch the subtopics given in each phylum. For example, in Protozoa, they have given the locomotion, nutrition, reproduction, protozoan diseases of man. In this thing, what is locomotion? What are all the locomotory organelles used by the protozoans, and what are all the methods, which organelle is used by which type of organism? That is very important. For example, there are different types of locomotory organelles are present, like pseudopodia, that is false feet. Today we are going to discuss protozoa and its locomotion. I am going to elaborate in that. Pseudopodia is the locomotory organelle, flagella is the locomotory organelle, cilia, and by using all these things, they are going to perform several types of locomotory movement. Okay, and what about this nutrition? There are different types of nutrition followed by this protozoa, that is holozoic, holophytic, saprozoic, and parasitic. These are all the different types of nutrition, and you have to know which organism prefer to acquire the nourishment with which type of nutrition. Examples is very much important. And what about this reproduction? There is asexual reproduction and a sexual reproduction is there, as we know, but protozoa prefers which type of reproduction, and in case of that reproduction, what are all its types? In case of asexual reproduction, there is a binary fission, there is a multiple fission, there is a schizogony, everything is there. And we have to understand which type of reproduction is followed by paramecium, which type of new reproduction is followed by amoeba. The examples is important. Next, when it comes to the protozoan diseases, you have to read what are all the protozoan diseases. So whatever be the diseases they have given, you have to read name of the disease, name of their organism, that is with pathogen. Which pathogen is responsible for causing this type of disease, and in case if it caused the disease, what is their host organism they are preferred to live, and what is the primary host and what is the secondary host, and in case if it affects, which larval stages is going to affect that particular host. And we have to read what are all the larval stages, and if in case they are going to perform the sexual reproduction and asexual reproduction, we have to read what is the sexual reproduction is takes place in which organ of that particular host. Asexual reproduction is takes place in which organ of the particular host. For example, we can take malaria. Malaria will undergo they complete their life cycle by involving in two different hosts, that is one is mosquito and the human. We have to know that malaria, that mosquito is just a vector, but the actual disease causing agent is the virus, and that virus complete their life cycle by entering into the mosquito, by entering into the salivary glands of mosquito and the humans, and the humans by entering into these two hosts, they complete their life cycle. So we have to know nook and corner about the protozoan diseases, and after it causing the diseases, what is the treatments are available, and what we have to prevent to completely avoid those diseases. We have to focus on these things. For example, malaria, trypanosoma gambians, schizo, we are going to discuss everything. Okay, next, porifera. In porifera, they have given the canal system of porifera, skeletal system, and reproduction in sponges. We have to know. See, for each phylum, there has an unique features. For example, in case of porifera, it is canal system. In case of echinodermata, there is a water vascular system. Like that, in case of arthropoda, there is, as the name suggests, a jointed appendages will be present in arthropoda. In case of annelida, segmented worms, they are said to be as the segmented worms. So each and every phylums, they have an unique features. We have to focus on those unique features. Like in the same way, porifera has the canal system in porifera. See, if you know, they have given the canal system in porifera. What we are going to read is what is canal system and what are all the types of canal system. and what is the pathway which when the water enters into the sponges, what are all the route they are going to prefer in those canal system. What is spongocell? Everything we are going to discuss when we read the canal system in porifera. Right. Next comes to the skeleton in the porifera. When we discuss about the skeleton in porifera, we are going to focus on the spicules and the spongin fibers. These spicules and spongin fibers, they are going to comprise the entire skeleton system of the porifera. We are going to read that. And reproduction in sponges. What are all the reproductions followed in the sponges? Okay, and examples for those reproduction. And next comes to the coelenterata. See, why a general characters and classification is very important as a very important means you have to know, if you know the characteristic features of coelenterata, we also come into a conclusion why corals is placed under the coelenterata. If we know the general characteristics features of echinodermata, we can also able to understand why starfish is placed under the echinodermata. So for that reason, if you know the general characteristics, whatever the examples they have given in the exam, you can easily locate that particular organism in the exact phylum. That is why reading the general characters and classification is important. Now, next comes to the helminthes. As I said before, this helminthes comprises two different phylums. Okay, and what is that? Platyhelminthes and nemathelminthes. Okay, in platyhelminthes, it is also called as the flatworms. In case of nemathelminthes or aschelminthes, they are said to be as the roundworms. See, common helminth parasites of man, they have given certain parasites like Taenia solium, Schistosomia species, Ascaris, Ancyclostomia, Oxyuris, Lova, Trichinella, Strongyloids. See, they have given a list of parasites. What we are going to focus is, take for an example, Taenia solium. What is the common name for Tediosolium? Straight away, they can pose the question in the exam. What is the common name? Porkworm is the common name of which parasite, which biological name? Like they can strike a question. Okay, so we are going to read what is the common name. Okay, and second one is what is the diseases they are going to cause to the host. In case they are going to cause a diseases to the host, which type of host they are going to prefer? In case they are going to cause a disease by entering into the host organism, what are all the types of host, which means what if they are going to complete their life cycle by involving into two hosts or the single host, that is a monogenetic life cycle or the digenetic life cycle. We are going to focus the primary host and secondary host. What is primary host and what is secondary host? Okay, and the most important thing is larval stages. What are all the larval stages undergoing by these type of parasites? In case the order of the chronological order of the larval stages is very important. For example, we can take this Taenia solium. What are all the larval stages? What after the Miracidium? What is the next larval stages? That order is very much important. And one more thing is which larval stages going to convert into an adult form. Okay, and that scenario is takes place in which host, that and all very important. And the most important thing is what are all the diseases, and in case if it causes any diseases, what are all the treatments and preventions are available. Okay, that is very important. Like in the same way, we are going to read all the parasites. That's all about the helminthes. Helminthes is totally parasitic in nature. So we are going to focus this life cycle and parasitism, and what are all the parasitic adaptations. In case they are going to cause a diseases to the host, what are all the adaptations they are undergoing? Everything will be dealt here. And Annelida. In Annelida, from this phylum, the concept of metameric segmentation will arise. Okay, see why? Remember, you have to know each and every point which is mentioned in the syllabus. And one more important thing I have forgot to say in the starting. See, you have to know each and every word in the syllabus. What are all the things mentioned in the general concepts? What are all the things? What are all the words which is mentioned in non-core data? What are all the words mentioned in the core data? If you want to memorize it, just memorize it. Why I am saying all these things is, see, even though you didn't touch the topic at all, if you know it is present in the syllabus, and if you know 10 to 15 points with respect to that particular topic, you can easily crack the question. My voice is not audible. From where I have to repeat? Am I clear now? Kindly, one more thing is, when you log into the class, just type your name and log in. Okay. Okay. Am I clear now? Okay. Fine. Up to where I have to repeat. See, okay, I'm repeating. See, why I am saying the syllabus is important means, even though you didn't touch the topic, even though you didn't remember what are all the informations I have sent in the class, if you know the syllabus, you can easily eliminate the option. For example, not audible. Just recheck your network connection. Okay, do it, do it. Okay, let me start. See, revising or reading the syllabus is very, very important. Why? Because if you know the each and every word, see, if you know what is acylomate, pseudocoelomate, and coelomate, and what is the meaning of the acylomate, and what are all the phylums which is placed under this acylomate, and acylomate is coming under which division? Acylomate is a subdivision, and this subdivision comes from the which division? If you know these basic things, you can easily answer the question. If they ask the question with respect to the coelom, body cavity, means whatever the question they can throw at you, you can easily eliminate the option. You can easily answer the question by eliminating the option. So, see, like that, where I have left. See, from Annelida, the concept of metameric segmentation will be arise. See, up to this Nemet Helminthus, I didn't even mention a single word like segmentation, that too metameric segmentation. From this phylum Annelida, the metameric segmentation, the concept will be arise. What is metameric segmentation means? The body is completely made up of a segments. That is why Annelida have a special name called the segmented worms. Okay, like this. Take for an example Peritema, that is earthworm. Okay, that body is entirely covered by the segmentation. This is called as a metameric segmentation. Each segment is destined to do different types of functions. Okay, that is called as the metameric segmentation. I'm going to explain all those things when I dealt with the coelom formation, coelom and coelomoducts. And one more thing they have mentioned in the syllabus under the Annelida is excretory system. What are all the excretory system or excretory organelle used by those segmented worms? That is nephridia, nephriostome. Everything will be dealt when I explain the excretory system. Next, arthropoda. They have given the mouth parts of insects, useful and harmful insects, metamorphosis in insects, apiculture, sericulture, and crustacean larvae. See, in arthropoda, they have given a mouth parts of insects. There are different types of mouth parts of insects is there. That is siphoning type, chipping type. There are different types of mouth parts inside. What you are going to focus, what we are going to focus are what are all the types of mouth parts and which insects following which type of mouth parts. For example, butterfly is following the siphoning type of mouth parts. Like that, you have to read for all those things, right? Fine. Next, useful and harmful insects. See, insecta plays a major role in all the phylums. Which phylum plays a secure a largest portion in the animal phylum? Can anyone say? In the 9 phylums, starting from the Porifera to Echinodermata, which phylum secures the largest area when compared to the other phylum? Quick. Nobody knows. See, Arthropoda securus major is the largest phylum. Oh, yes, Arthropoda is the largest phylum. That too, in that Arthropoda, there are different types of classes are present. In that Insecta, the class Insecta secures the largest class in under the Arthropoda. Because of this Insecta, Arthropoda is the largest phylum in the animal kingdom. Okay, and what are all the useful? Not all the insects are very useful. Some insects are very useful when it comes to the pollination. Not only pollination, they are going to helpful in the dispersal of seeds. Like that, many are useful, many are harmful. We are going to list out all those insects. And metamorphosis of insects. Metamorphosis of insect means their larval stages. Complete metamorphosis is there, incomplete metamorphosis is there. There are 4 different types of metamorphosis in insects is there. We are going to discuss what are all the different types of metamorphosis and what are all the other names. All the 4 types have another name, and we are going to focus on those names also. And what are all the examples? Which insect is going to follow which type of metamorphosis type? And another one is apiculture and sericulture. In case of apiculture, culturing of honeybees, we are going to deal with the culturing of honeybees and what are all the process they are going to follow in this apiculture and how they are going to harvesting the honeys. Okay, that and all we'll deal with apiculture. When it comes to the sericulture, what is sericulture? Culturing of the silkworm, and what are all the process they are undergoing in the sericulture unit and how they are going to culturing the silk from the cocoon. Everything will be discussed in the sericulture. And crustacean larva, they are the most questionable area among the, when compared to the all other topics given in arthropoda. This crustacean larva is very crucial because there are different types of crustacean larvae there. As I said before, when it comes to the larval stages, you have to know the order, that's chronological order, which larva is upgraded to which larva. That is very important. Alyma larva is the last stage of larva in the crustacean. Before that, phylozoma. Before that, zoea larva, nauplius larva. Everything will be discussed when it comes to the crustacean larva. Next, mollusca. Mollusca is said to be as the shelled animals. In that mollusca, they have given the respiration, torsion and detorsion, pearl formation, and pearl industry. Among all other topics, we are the most important topic. The most important topic is torsion and detorsion. What does see? As I said, each phylum has its own unique characteristic features. Like for Porifera, canal system, like Echinodermata, water vascular system. This mollusca, that too exclusively gastropoda. Gastropoda is one of the class under the mollusca that is going to follow this torsion and detorsion. We are going to discuss what is torsion and what is detorsion, what is the difference between the torsion and the coiling, and what is the significance of torsion, why these gastropods undergo this torsion, what are the significance. Everything will be discussed in torsion and detorsion. And pearl formation and pearl industry, I'm going to discuss completely about the pearl culture. Okay, and next the echinodermata. We see echinoderm larva. See larval stages wherever in the syllabus you are seeing the larval stage. You have not omit those topics. See, as I said, if write the syllabus in what I am suggesting is not for this 4 unit, for all the 11 units you have to write down the syllabus. And for each, for example, in case of crustacean larva, for arthropoda after completing all the syllabus. See, once every syllabus, every topic in the syllabus have been completed, what is your job to do is you have to write down the syllabus. And for example, take for example crustacean larva. You have to read what are all the crustacean larva. You have to you do not change the order. You have to write in your notebook. For last minute revision, it helps a lot to you. Okay, so don't miss that. And like in the same way, echinoderm larva. What are all the larval stages which is going to undergo by this echinoderms, and what are all the class will follow which type of larva? For example, bipinnaria larva is followed by which class under the echinodermata. All these things you have to remember. And water vascular system, which system is absent in echinodermata, and in replace of that, which system, this water vascular system is present in echinodermata. That will be the questionable area. Say for example, in echinodermata, excretory system is not present in there. Some of the other systems are not present in here. I'm going to elaborate in detail whenever I am discussing the echinodermata. In place of excretory system, this water vascular system is present. I'm going to explain what is water vascular system and what are all the types, what are all the parts which is involved in the water vascular system, and each part is destined to do which type of function. Everything will be discussed in the water vascular system. That's all about the non-chordata. Now comes to the chordata. As I said before, general characteristics and classification is very much important. Whatever be the phylum and whatever be the class, you have to read the general characteristics. Okay, I'm going to explain. I'm going to start with origin of, after completing the general characters and classification, I'm going to focus on the chordates origin. We know chordates organisms are evolved from the non-chordates, and I'm going to prove how this non-chordata is going to evolve into the chordates animals and their evolution and their similarities. Just answer my question. Which organism is said to be as the connecting link between non-chordata and chordata? Which a phylum or example, whatever. Just tell me which organism is the connecting link between the non-chordates and chordates. Anybody remember? Okay, let me say, see, very good. Balanoglasses. Balanoglasses comes under which phylum? Hemichordata, right? See, from non-chordata, chordata will be evolved. In that, before non-chordata, there is a connecting link that shares a common features which is present in non-chordata, as well as it shares the common features from the chordata. That will be considered as the hemichordata. It is partially considered as a chordate, partially considered as a non-chordate. The best example is balanoglasses. Very good. Now I am going to explain that in the phylogeny and affinities of hemichordata and retrogressive metamorphosis. Okay, and the second one is I'm going to compare the systems, digestive system, circulatory, excretory, respiratory system of all the vertebrates, how they look like and how unique they are. And the second one is physiculture in India. What is physiculture, and what are all the types of physiculture, and what are all the things they are following in this type of physiculture. And one more thing is common edible fishes of Andhra Pradesh. They didn't focus anything about India, common edible fishes of India, but they have focused only in Andhra Pradesh. So you have to read. It is very easy for you to remember all those edible fishes, not all the fishes, that too edible fishes, particularly in Andhra Pradesh. So it is very important. You can easily crack the questions when they have arised from this edible fishes of Andhra Pradesh. Okay. Now, origin evolution of the amphibia, pedogenesis and neoteny. See, you have to read what are all the general characters and classification of amphibia. After reading that, you have to read what is pedogenesis, what is neoteny. See, neoteny and pedogenesis will be followed and only in the amphibia. What is axolotl, everything will be discussed in the amphibia. Next, important snakes of in the reptiles, they you have to read the general characters of reptiles and classification of reptiles, and you have to list out the important snakes of India, and you have to read what is poisonous snakes and what are all the list of poisonous snakes and list of non poisonous snakes. And after reading that list, you have to read what are all the difference between these poisonous and non-poisonous snake, how we can identify whether it is poisonous or non-poisonous by seeing those outer structure. We have to read those things, those differences. And I am going to explain the evolutionary history about the dinosaurs and most important facts about this dinosaurs. Next, flight adaptation and migration in birds. When it comes to the apes, you have to read the general characters and classification of the apes and what are all the flight adaptation. For this flight adaptation, which muscle is important for the birds they are going to adapt for their flight. Everything will be dealt in the flight adaptation and migration in birds. In which season they are going to migrate, for what reasons they are undergoing migrate, and some of the factual information is very important. Which bird is very smallest, which bird is very largest, and which is the sharpest, and which shows the longest distance of migration, and which shows the shortest distance of migration. These facts will be also an important. It is not very important, but you have to know these things when you read the migration in birds. And next one is Archaeopteryx. What is the speciality of Archaeopteryx, why it is considered as a fossil evidence, everything will be discussed when I explain the Archaeopteryx. Poultry in India, what is poultry, and if we know the name poultry, what we are going to think is just just we remember only the chicks. The poultry is not only the dealt with the culturing of the chicks, but also different types of birds that are collectively called as the poultry. We are going to discuss what are all the birds they are going to cultivate or culture in the poultry, that too in India. Okay, next, adaptive radiation and dentition in mammals. See, there are different types of adaptive radiation followed by the mammals. For example, cursorial, fossorial, running, leaping, different types of adaptive radiation followed by the animals according to their survival, according to their environment. We have to know from which common ancestor these all other evolution have been made, and which type of adaptation followed by which type of mammals. That examples is an important. And the dentition in mammals, when it comes to the dentition in mammals, you should know what is the difference between teeth and the dentition, and what is mean by dentition. And one more important thing is I have a list of mammals, and for that list of mammals I also having the dentition formula. The dental formula plays a crucial role when it comes to the questionable area. So you have to focus the dental formula in the mammals. Okay, that's all about the chordates. And now histology. Histology is nothing but the study about tissues. In that the tissues, what are all the tissues? Or I have one PDF, which means what, one single page of a flow chart. If you read those flow chart, you can easily whatever the question they have asked in the exam, in that single flow chart everything will be there. You have to that perfect that example, there are how many different types of epithelial. For example, epithelial tissue is there, connective tissue is there, and many different and many tissue is there, majorly three types. And in that epithelial tissue, what are all the types of epithelial tissues? The squamous is there, cuboidal, columnar epithelial tissue. In that squamous, which are all the organs which comprises this squamous epithelial tissue? Which are all the organs which comprises this cuboidal and columnar epithelial tissue? All these things are very very important. And I have that single flow chart, and you have read this, and whatever the question they have asked in the exam, you can easily answer the question when you read this. Okay, that's all about the histology. That's all about the syllabus explanation. Yesterday, one person asked about explaining the syllabus, so I have completed now. If you have any doubts, you can ask me now. Any doubts? Radhika, Devi, Lalita, Ziomi. Any doubts? Okay, what about other two? Another one. Kindly, when you once again, I remember you when you log into the class. Only today, I'm going to take the class in the Zoom. From tomorrow, I'm going to take it in the app. So don't worry. Sorry for the misconvenience. Okay, Xiomi, are you clear now? Okay, fine. That's all about the syllabus explanation. Now, I'm going to explain few questions and you have to answer for those questions. In yesterday's class, I have completed about the classification, general characteristics of invertebrates and classification of invertebrates. Everything I have explained yesterday. I'm going to ask a few questions. You are going to answer now. You have to answer very quickly. Don't take much time. I have to finish Protozoa and its locomotion by today. Okay. Uh, what is enema and anima? What does enema? This ENAIMA. Who classified the animals, the two animal, in based on their RBC? Anything you have to say. Okay, very good. And what type of classification is this? Enema and anima is put forth by which scientist? Aristotle system of classification. Okay, what about the other two? Kindly answer. See, Aristotle system, very good. Aristotle system of classification, he classified animals based on the blood and based on habitat. One is enema and anema. You have to read this classification and mm, okay. The most accepted system of classification is, what is the most accepted system of classification? Nobody has revised yesterday, I think. Is it phylogenetic? Most accepted system of classification is whose system of classification? I'm asking the scientist. Yesterday, we have seen Ernest Haeckel three-domain classification, Carl U six-kingdom classification, R.H. Whittaker traditional five-kingdom class system. In these three, which type of classification is mostly accepted? We take a system of classification. See, whatever the thing I have explained in today's class, you have to revise on those same day. Why? Because in the next day I'm going to ask many questions and you have to answer. If you read like this means it is very easy for you. Okay, at the end of the day. Okay. Agaricus is the example of which kingdom? Agaricus is an example of which kingdom? First, fungi. Agaricus is the species of mushroom. Okay, and fungi is the example of this kingdom. Okay, in hierarchical taxonomical classification, which is the basic unit in this hierarchy? Which is said to be as the basic unit? I said in the PPT, no, you have to revise in that. From today, recorded classes will be available. Don't worry. But you have to revise the PPT. Okay. Okay. Okay, just answer for this question. What did I ask? In taxonomical classification, which is considered as the basic unit? Fast, fast. Nobody knows. See, basic unit is the species. Okay, domain is a broader area, eukarya. Yes. Why you are thinking for the simple question, domain is eukarya, kingdom, animalia. In that many phylum is there. In that many class. In that many order, many family. In that many one genus contains many species, and that species is considered as the basic unit in the hierarchical taxonomical classification. Okay. What is the difference? I mean, what is the only similarity which is placed under the invertebrates? Which is what is the only similarity for the organisms which is placed under these invertebrates? Starting from the worms, butterflies, sponges, corals, snails, crabs, starfishes, they are the different range of animals. Yes, they are different range of animals, but they are connected by only single similarity. That too, they have no notochord and no vertebral column. The only thing which unite all these organisms under the invertebrate is no absence of vertebral column. Very good. Next, we know the what is the difference between the invertebrate and non-chordate? What is the basic difference? Just tell me that single phylum which makes a difference between this invertebrate and notochord. Sorry, invertebrate and non-chordates. Very good. Protochordates. If we eliminate the protocordate from the invertebrate, we can get the non-chordate. But generally, we are saying non-chordates and invertebrates are same, but they are not actually synonyms, right? Slightest difference between these two. Now, okay, you have to read. I can ask the phylum, for example, for these phylum, but I think you won't remember. So you have to read this classification and which phylum is placed under which category. You have to read this. And okay, these are all about. Okay, what is the common name for Mastigophora? What is the common name for the Mastigophora? Mastigophora is a superclass under the Sacromastigophora. I have said yesterday there is a common name for Mastigophora. What is it? Very good. Flagellated protozoans. They are said to be as the flagellated protozoans. What is the other name of Sarcodina? They are said to be as the amoeboid protozoans. Okay, we have stopped up to this. Okay, now I'm going to explain whatever the thing I have explained yesterday, I'm going to explain now. See, what is Mastigophora? Mastigophora is also called as the flagellated protozoans. Why means they have a flagella in it. This is considered as the flagella. See, just focus on the diagram. See, this is called as the pellicle. Pellicle is the outer covering which is found only in the some protozoans. Pellicle is the characteristic features of the protozoans. This is the pellicle. See, this mastigophora or flagellated protozoan, they are parasites or free living. Whether they are depend on their host organisms to acquire their nourishment or they can live independently. Okay, and they using flagella for locomotion. As I said yesterday, these mastigophora, sarcomastigophora, sporozoa, nidospora, ciliophora, mainly one of the main criteria they have been classified based according to it is locomotion. Based on the locomotion, they are separating this subphylum. That is why Mastigophora uses flagella for its locomotion. They have a thick cuticle or its pellicle. See, freshwater forms have freshwater forms. The flagellated protozoans, they whether they have if they are going to prefer the freshwater, they have a contractile vacuole. What is the function of the contractile vacuole? Means it is the main function of contractile vacuole is osmoregulation. It is considered as an osmoregulatory organelle. And the reproduction followed by the organisms which is placed under this flagellated protozoans. What are all the things as considered as a flagellated protozoans? Their reproduction is by binary fission. Reproduction is by binary fission. Example is a game changer. You have to remember the examples. Trypanosoma, Trichomonas, Giardia, Leishmania, everything is an example of flagellated protozoans. And Sarcodina, as I said before, Sarcodina is also called as the amoeboid protozoans. So obviously, what type of locomotory organelle is used by those amoeboid protozoans? Exactly, pseudopodia. Amoeboid. When you remember the amoeba, the locomotory organelle, they are going to use is pseudopodia. That is false feet. They are looks like a feet that is not actually a well-developed organelle. Just the exclusion of the outer cell wall. See, they are going to live in the fresh water, sea water, or the moist soil. This pseudopodia is necessary for its locomotion, and this pseudopodia is not only not only meant for its locomotion but also to capture their prey. Okay, for their food acquiring organelle. And there is no definite shape. Right from the beginning, we are reading, right from the childhood days, amoeba doesn't have a no definite shape. It is irregular in shape. As it is irregular in nature, there is no pellicle is present here. Pellicle is absent in the amoeba protozoans. And see, contractile vacuole is present. See, contractile vacuole is an osmoregulatory organelle, so it is definitely going to present in the amoeboid protozoans. Reproduction is done by the binary fission and cyst formation. Examples are amoeba and entamoeba. Okay, you have to remember the example. And next comes to the sporozoa or sporozoans. See, sporozoa are said to be as the 1 minute. Please mute yourself, Xiomi. Good. Okay. They are said to be as the endoparasitic. Why? Because they are going to live inside the parasitic. See, when it comes to the parasitic, what is meant by parasitic? They are definitely going to depend on the host organism for their nourishment. And there are two different types of parasitism. One is ectoparasites and another one is endoparasite. Okay, what is ectoparasite? The parasite which is going to live on the outer body of the host. But endoparasite means they are going to live inside the body of the host. So sporozoa or the sporozoans, they are endoparasitic. The best example is Plasmodium. Okay, and they don't have a specialized organ for locomotion. It is a spore and it is one of the best method of asexual reproduction, and this spores doesn't have a specialized organelle for the locomotion. When the as I mentioned before, this sporozoans doesn't have any locomotory organ. Okay, see here the pellicle is present which has a subpellicular microtubules that helps in movement. If it doesn't have any locomotory organelle, who is helping them to move from one place to another means this subpellicular microtubules. We know that microtubules, the cytoskeleton, we know microtubules is the one going to help the cell in the motility. Okay, so for sporozoans, as it lacks its locomotory organelle, these subpellicular microtubules is the one helps the sporozoans to move from one place to another. And this reproduction is done by the sporozoite formation. As I said before, this sporozoa is one of the main main important asexual reproduction followed by the sporozoans. Examples are Plasmodium, Myxedium, Nosema, Globidium. Examples you have to remember in tomorrow's class. I'm going to ask you. Definitely. See, Ciliophora are ciliated protozoans. The best example is paramecium, Vorticella, and the Bilantidium. Whatever the examples you remember or not, just remember the paramecium at least. Okay. See, they are aquatic. They are aquatic and move actively with the help of thousands of cilia. You can find in the paramecium, the entire body is covered by the cilia. Okay, and these cilia is the one going to help them in the locomotion. They have a fixed shape due to covering of the pellicle. Because of this pellicle, they are looks like a certain a definite shape. Unlike amoeba, amoeba is irregular in shape because of the absence of pellicle. And for if the presence of as the paramecium has the pellicle, it looks like a definite shape. It acquires the definite shape. So they may have the tentacles. What does tentacles means? They are used for grasping and feeding the organisms or the nourishment or the nutrients, whatever it is. The tentacles is helps by the used by the organisms for grasping and feeding. Contractile vacuole is present, obviously osmoregulation is going to happen. What is contractile vacuole, what is its function, everything will be explained in the today's class. Some species have an organ for the defense called the trichocyst. What does trichocyst mean? This is the defense organelle. What is the defensive organelle present in paramecium or present in ciliated protozoan is trichocyst. They are going to use it against the predators. Next, they move with the help of cilia, and we know that. And movement of cilia also helps in taking the food inside the gullet. As I said in the yesterday's class, cilia is not only meant for the locomotion but also for acquiring the nourishment. You also know what type of nutrition they are going to follow. Transverse division also from the cyst. See, consider this is the paramecium. Transverse division means this upper part is going to converted into the new paramecium, and this lower part is going to converted into the newer paramecium. This is called the this cut across the animal or across the organism, that is said to be as the transverse division. Okay, that's all about the locomotory organelle. Now only we are entering into the process called locomotion. See, what are all the locomotion in protozoa? Sorry, these are all the class of the protozoa. Now we are going to see what are all the locomotory organelles. First one is pseudopodia. In that pseudopodia, there are four different types: lobopodia, philopodia, reticulopodia, axopodia. These are all the four different types of the pseudopodia. As I said before, pseudo means fall, podium means feet. Looks like a false feet, and they are how they are moving means by streaming of the cytoplasm. What is the streaming of cytoplasm means? Cytoplasmic streaming. It is said to be as the cytoplasmic streaming or the protoplasmic streaming, or it is also called as the cyclosis. Okay, it is also called as the cyclosis. What is mean by streaming of cytoplasm means flow of cytoplasm inside the cell driven by the forces from the cytoskeleton. Okay, just the movement of cytoplasm, that is said to be as the streaming of cytoplasm. And next one is what are these are this is meant by this is meant to what is called as the pseudopodia. And the different four types of pseudopodia are: first one is lobopodia, and the best example is amoeba. Okay, why it is said to be as lobopodia means the pseudopodia present in this type of animals are looks like a lobe. That is why they are called as the lobopodia. Example is amoeba. See, they lobe-like pseudopodia with broad and round ends, composed of both ectoplasm and the endoplasm. Lobopodia moves by the pressure flow mechanism. Pressure flow mechanism. What is meant by pressure flow mechanism means in the sense it has to maintain the highest pressure for the locomotion. It has to put more pressure, more energy they have to use for this uh, movement. Okay, and the second one is philopodia, and the best example euglypha. See, what is the difference between this lobopodia? The name comes from its structure. As this organisms looks like a lobe, they are said to be as the lobopodia, and this philopodia is looks like a filamentous pseudopodia. It is called as the philopodia. It has a pointed tip. See, in lobopodia, it has both ectoplasm and endoplasm. In philopodia, it has only the ectoplasm. It has a branch and forms simple and complex network. In reticulopodia, just remember the example, Globigerina. It is also called as the rhizopodia or mixopodia. See, it has filamentous in nature. These are all the filament. How this filament is looks like, how this filament is look like. It is straight pointed tip. It is looks like this, but here it is looks like this. This is also a filament, this is also the filament, but a structure is slightly different. From this structure, the name reticulopodia will arise. See, branched, interconnected profusely to form from the networks. It displays the two-way cytoplasm. The most important questionable area is which type of pseudopodia will displace the two flow of cytoplasm. What is two flow of cytoplasm means? See, it can move in both both clockwise and the anti-clockwise direction. Among the many smaller vacuoles, see this movement is mediated by the motor proteins. So whatever be, whether it is a streaming of cytoplasm, whether it is the two-way flow of cytoplasm, everything will be under the control of what? Motor proteins. That too in the which motor proteins? Motor proteins of the microtubules. You shouldn't forget this. And this movement, what is generally the two-way flow of cytoplasm means it can able to move both clockwise and anti-clockwise direction. Okay, that's all about the reticulopodia. And axopodia, actinosporium and actinophores, they are heliozoans. What is heliozoans mean? These are the animals can feed by the process of phagocytosis. They are said to be as the heliozoans. And what are the examples of those heliozoans means actinosporium, actinophytes. This can be asked in the question. See, like reticulopodia, this axopodia also displays the two-way flow of cytoplasm, which means the cytoplasm, the ability of movement of cytoplasm in both clockwise direction and the anti-clockwise direction. That is called as the two-way flow of cytoplasm. See, more or less straight pseudopodia radiating from the surface of the body. Each axopodia contain a central axial rod in which covered by the granular and adhesive cytoplasm. When it comes to axopodia, just remember the diagram and remember the unique feature of reticulopodia and axopodia and remember the example. This is sufficient. That's all about the pseudopodia. Next comes to the flagella. The best examples of the organisms which is using the flagella as its locomotory organelle is euglena and the trypanosomia. See, in a flagella, there is a thread-like projections on the cell surface, and it is consist of elongate, stiff axial filament, axonum, enclosed on the outer sheet. Axonum. See, just see the structure. This is called as the microtubules. It is present in the 9+2 arrangement, which means 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, 9 microtubules, which means 9 paired fibers in the central. These are everything is the microtubules. Because of the movement of this microtubules, the flagella can move, and because of the movement of flagella, the entire body of the organisms will move. This is how it performs. See, this is the outer microtubule doublet. Always the microtubule which is present in the flagella occurs in a pair only. That is why it is called as the outer microtubule doublet, central microtubules. Everything will be occur in pairs, 9+2 arrangement. This is also an important. Because of this movement, the flagella, because of this microtubular movement, the flagella will move, and because of the movement of flagella, the entire body of the organisms will move. That's why we are saying flagella is meant for the movement. Next, see where this are arising means basal body. This basal body structurally identical to central. From this basal body, this microtubule will arise. Next, cilia. The example of cilia is Orticella and the euplots. See, same. Just like flagella, it is also occurs in the 9+2 arrangement. See, this is all the questionable area. Don't take it lightly. Okay. See, cilia are the 9 paired peripheral fibers. See, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 paired peripheral fibers and the 2 central fibers. They are all embedded in the structural matrix. They are all embedded in the matrix in between the outer and inner fibers. See, there are different rings are present here, and one of the most important thing, which motor, as I said before, these microtubules are associated with the motor proteins. Always the movement is caused by the motor proteins of the microtubules. And what is the name of the motor protein which helps in the movement of the microtubule means dynein. This can be asked. Dynein is the motor protein. Nexin is the motor protein. You can easily answer when they ask the question. If you know the structure, you can easily answer. 9 peripheral fibers, the 9 peripheral microtubule doublet and the 2 central ring. Okay, the motor protein is dynein. This is considered as the inner dynein. This is considered as an outer dynein. These two are connected by the nexin. This is called as the central link. This is called as the radial spoke. Okay, fine. See, 9 radial spokes are present. Totally, how many radial spokes are present? 9 radial spokes are present. They are going to regulate the dynein motors for motility. See, generally, if we say an overview, just microtubule is meant for locomotion, but for microtubule movement, which protein is helping? That is called as the motor protein, that too dynein. Okay, and for this movement of motor protein called dynein, which is helping here, radial spoke is helping here. This 9 radial spoke, this is the one going to regulate the dynein motors for motility. Okay, that's all about the cilia. And next, pellicular and contractile structure. Example, euglena and trypanosoma. In the pellicular contractile structure, see, when you remember the pellicular and contractile structure, you have to remember the word myonems. Again, the cytoskeleton microtubules will come into picture. This pellicular contractile structure, which is called as the myonems, this is the one aids in the locomotion. Okay, by just by contraction and relaxation of the muscles. Okay, see, this contraction, this myonems has many protein filament that can contract. This myonem is helps in the locomotion by just by contraction and relaxation. See, in myonems there are different types of proteins. That is the one helps the myonems to perform the contraction and relaxation, and that motor proteins, they are going to contract and relax only they are going to expose with certain ions. See, this myonems has many protein filaments that have contract when exposed to the calcium ions. See, when they exposed to the calcium ions, they are going to contract. Remember this. That's all about the locomotory organs. Up to this, locomotory organs will be completed. Now, what are all the methods of locomotion? The most important type of locomotion is solgal, that is amoeboid movement. Then theory which explains this amoeboid movement is solgal theory. There are 4 different types of locomotion is there. One is amoeboid movement, another one is flagellar movement, another one is undulating movement, another one is simple conical gyration movement, and ciliary movement, metabolic movements. These are all the locomotory movements. And the first one is amoeboid. In the 4 method, the first one is amoeboid movement. See, to explain this amoeboid movement, we should know one theory that is called as the sol gel theory. See, generally, amoeba cytoplasm has two layers. One is sol and gel. Okay, amoeba layers have amoeba cytoplasm. The cytoplasm is considered as the has the comprises of two layers. One is plasmosol and plasmogel. See, the inside it is called as the plasmosol. This is called as the plasmosol. The outer thick blue color is called as the plasmogel. It is made up of two layers. See, this layer is considered as the plasmosol. This entire area is called as the plasmosol, and this thick blue color it is called as the plasmogel. Okay. Now, this plasmosol is said to be as the viscous fluid. See, this is very important theory. Try to remember. Listen carefully. This plasmosol is said to be as the viscous fluid. This plasmogel is said to be as the gelatinous semi-solid. Okay, see, just remember this amoeba cytoplasm has two type of layers. One is plasmosol and plasmogel. Plasmosol is said to be as the viscous fluid, and the plasmogel is said to be as the gelatinous semi-solid. Okay, during a locomotion, this amoeba moves these gelatinous plasmogel. See, this is the plasmogel, this entire blue color. During the locomotion, when the amoeba, if the amoeba wants to move, means first thing they are going to do is the movement of the plasmogel. The gelatinous plasmogel forms a rigid structure at the boundary. See, this plasmogel forms the rigid structure, which means what? They remain stable. They are rigid, means they are not flexible. They are not going to move. Okay, this plasmogel forms a rigid boundary at the boundary of the cell, and the fluid plasmosol, the plasmogel is rigid. They stand just like that. They forms a rigid at the boundary, and this plasmosol will move and extends forward. This will be stable and this will be move. Okay, and next, the plasmosol and extends forward means the animal will move forward. Next, the plasmogel then moves to the zone of isolation. See, after that, see, initially when the amoeba wants to move, this plasmogel forms a rigid structure. Only the plasmosol moves and extends forward. And after that, this plasmogel will move to the zone of isolation. Okay, zone of isolation, where it becomes the plasmosol. See, this plasmosol enters into the zone of isolation where the plasmogel is converted into the plasmosol. Okay, after that, they are again they are going to move into the zone of gelation. After that, they are enter into the zone of gelation where it becomes to the plasmogel. Zone of isolation means the place where the plasmogel is converted into the plasmosol, and the zone of gelation means where the plasma gel is again converted into the where the plasmosol is converted into the plasmogel. Okay, this is called as the sol gel theory. In simple terms, we can say that their movement is induced by the converting the viscosity or rigidity. See, initially this plasmogel is viscous in nature. Plasmosol is viscous in nature, and the plasma gel is semi-solid in nature, gelatinous semi-solid. And this conversion of viscosity or rigidity of the protoplasm within the cell, this conversion is aids the amoeba in the movement. Okay, that's all about the method of locomotion. First movement, amoeboid locomotion. See, this amoeboid movement is the characteristics of all Sarcodina. As I said before, Sarcodina is called as the amoeboid protozoan. So all the Sarcodinas follows the amoeboid movement. And certain mastigophora and the sporozoa. Locomotory organelle used by these organisms is pseudopodia. just by the streaming flow of the cytoplasm, just by the conversion, their viscosity and rigidity, they are helps in the movement. Okay. Locomotion is possible only over the surface. It is not a deep movement. It's just a it's a movement. Okay, not a only over the surface. Depends on the active contraction of the ectoplasmic tube at the posterior end of the body. This is the posterior end with zone of solution. Because of the movement, because of the streaming of cytoplasm, they are undergoing into the movement. Okay. This leads to the endoplasm to flow forward into the expanding pseudoplodium. What are all the things I have explained here? Here it is not in detail. Just remember what I have said. See, the process involves, this is an important area. See, the process involves the xylation at the posterior end and the gelation at the anterior end. Okay, this will be the question. And this sol gel or the change of viscosity theory, is put forth by Mast and the Panton. Okay, just remember the name. I'll ask the tomorrow's class. See, that's all about the amoeboid movement. Next, the flagellar movement. Flagellar movement, you should know, amoeboid movement is followed by the sarcodina and some of the mastigophorans and some of the sporozoans. Like in the same way, flagellar movement is followed by the mastigophora. As the name suggests, mastigophora is also called as the flagellated protozoans. And this flagellar movement needs a liquid medium for its locomotion. There are three different types of flagellar movement. One is paddle stroke, another one is undulating motion, another one is simple conical gyration movement, and the first one is paddle stroke movement. See, paddle stroke movement has two different types. One is the effective downstroke. Just make a note of it. Effective downstroke. Effective stroke means the animal will move downwards. Downstroke is considered as a downstroke, and the recovery stroke. Recovery stroke is considered the relaxed state of the movement. See, this effective downstroke, this effective stroke will produce more thrust in the backward direction. More thrust in the backward direction. As a result, the animal will move forward. You have to remember, in effective stroke, the animal will move forward. Okay, the animal will move forward in the effective stroke. In case of a relaxed recovery stroke, the flagellum come back to its original position by its backward movement without any resistance. Okay, effective stroke means forward movement and the recovery stroke means a relaxed movement. This is called as this two stroke is comprisedly called as the paddle stroke movement, and this paddle stroke will be discovered by two different scientists, Ulela and the Christman. Okay, just read these things. And undulating motion. Undulating motion. Undulating motion is like a wave-like undulations. The animal will move from one place to another by producing the wave-like undulations of the flagellum. If the undulation passed from the tip to base, see, just write a note of it. If the undulations, if the wave-like undulations pass from the tip to base, the animal will move backward. Sorry, the animal will move forward. If it is a tip to base, the animal will move forward. Okay, tip to base, the animal will move forward. In case if the undulations pass from the base to tip, if the undulations pass from the base to tip, the animal will move backward. You have to remember these two important differences. See, when the undulations pass from tip to base, the animal will move forward. If the undulations pass from the base to tip, the animal will move backward. If the undulations are spiral, they cause the organisms to rotate in the opposite direction. Okay. Next, simple conical gyration movement. What is a simple conical gyration movement means? This movement was discovered with the help of axonemal fibers. What is axoneme? Axoneme is nothing but that microtubules. They axoneme because of that microtubules. Here also in this type of movement, microtubules plays an important role by the axonemal fibers. Axonemal fibers are involved in this type of movement specifically. And this microtubule doublet, when this microtubule doublet, say 9+2 arrangement, outer peripheral microtubule doublet, inner central microtubule pair. When this microtubule doublets slide past each other, cause the entire flagellum to turn like a screw, like this, turn like a screw. This action will pull the animal forward through the water with the spiral rotation as well as the gyration. Okay, and you have to remember, for this simple conical gyration movement, ATP is necessary. This cross bridges are formed and energy utilized for this process is ATP. That's all about the flagellar movement. And the next one is ciliary movement. In the ciliary movement, the best example is paramecium. Paramecium mainly moves mainly occurs by the movement of the cilia. It can be moved both forward as well as the backward. Cilia can move both forward and as well as the backward movement. If the animal move forward, if the animal move forward, the cilia moves from head to tail direction. Head to tail direction. See, this is the paramecium. The entire body is covered by the cilia. Consider this as the head region and this as the tail region. If the animal move forward, the cilia will move from head to the tail region. Then the animal will move forward. Like that, if the animal moves backward, the cilia will move tail to head, just in the opposite direction. You just remember, if the animal move forward, the cilia will move from head to the tail region. If the animal will move backward, the cilia will move from tail to the head region. Okay, this ciliary movement is defined as a rhythmic movement. Okay, this ciliary movement is said to be as a rhythmic movement that results in the fluid or the cell movement. And like flagella, it also needs a liquid medium for its locomotion. See, as I said, this ciliary movement is called as the rhythmic movement. Why I said this as a rhythmic movement means this shows two types of rhythms. First rhythm is synchronous rhythm and metachronous rhythm. That is why it is called as the rhythmic movement. One is synchronous and the metachronous. See, synchronous movement means when all the cilia move at the same time in the transverse direction, then it is called as the synchronous rhythm. See, I repeat, just see my statement, what I am mentioning. In synchronous rhythm, all the cilia, move at the same time in the transverse direction. This is the transverse direction. All the cilia will move at the same time in the transverse direction. Then it is said to be as the synchronous rhythm. You can find the video in the YouTube and you can see how the synchronous rhythm will look like and the transverse movement. And in case of metachronous rhythm, when cilia moves one after the other in the longitudinal, in case of a transfer movement, the animals will move like this. In case of longitudinal, the animal will move like this. The animal will move like this, or the animal will move like this. In case of a transfer, the animals will move like this, upward or downward. Okay, that is synchronous and metachronous. You have to remember, in case of a synchronous rhythm, the cilia will move at the same time in the transverse direction. In the metachronous rhythm, all the cilia moves one after the other in the longitudinal direction. Okay, that is called as the metachronous rhythm. Clear? Okay. And another important thing is, the ciliary movement is coordinated by the infraciliary system. See, this ciliary movement is coordinated by the infraciliary system. In connection with this infraciliary system, you need to know about the kinetosome and kinetodesmata. Okay, kinetosome. There is kinetodesmata and kinetosome. These are all the major important thing in the infraciliary system. In case of kinetosome, these are all the kinetosomes. See, this is the kinetosomes. This kinetosome This kinetosome, it is also called as a basal body. See, this is the kinetosome. This is also called as the basal body. Clear? This is the basal body or the basal granule. It is found at the base of cilia. See, this is the cilia. See, this is the cilia. This is the basal body where the cilia have been arise. See, this basal body is also called as the kinetosome. And what is kinetodesmata? Kinetodesmata, it is a strand running from the kinetosome. It is the kinetosome or a basal body. From this, many strands have been arised. That is called as the kinetodesmata. This red color line, it is said to be as the kinetodesmata. From kinetodesmata, the basal body is arising. From this basal body or the kinetosome, the cilia will be emerged. This is the infraciliary system. See now, these are all the parts present in the infraciliary system. Together in infraciliary system, together with the neuromotor system, helps in the coordination of the ciliary movement. Because of all these coordinations, cilia can move, cilia can take the animal from one place to the another. This is about the ciliary movement. And what about the metabolic movements? As everyone knows, protozoan has a pellicle in the ectoplasm, and this pellicle is composed of the proteinaceous stripes. It was supported by the dorsal and ventral microtubules. Okay, when these stripes slide past each other, the movements can be exhibited. So this, putting it simply, see, it shows a gliding or wriggling or peristaltic movement. These are all the another name of this metabolic movement. Okay, why it is also called as the Gregarine movement? Gregarine, it is also called as the Gregarine movement. Why? Because it is seen in the organisms called Gregarines. Contractile myelins or the microtubules present in their pellicular walls are responsible for this type of movement. Okay, just because of the movement of the microtubules, again all the doublet in the central tube, peripheral microtubules, and because of the slide path of each microtubules, the flagella can move, and that because of the movement of flagella, the entire organism will move. The same thing will be seen in the metabolic movements. See, what does Gregarine means? Gregarines are the subclass of the phylum Sporozoans. Okay, they are the parasites in the digestive tract of the insects, crustaceans, earthworms, and also in some of the organisms. Okay, that's all about the locomotion in the protozoans. Let me show you some of the questions. You can answer this. The functions of contractile vacuole is increased from. Okay, I'll post it in the WhatsApp group and you can answer the question. Okay, what subcellular physiological division of labor occurs in? Answer for this question. Subcellular physiological division of labor occurs in. Okay, subcellular physiological division of labors means what? Each organelle is destined to do different function. For example, food vacuole is for digestion, contractile vacuole is for osmoregulation. But in protozoa, it is not very well developed. That is why it is called as the subcellular physiological division of labor. So the answer is protozoa. And see, cilia in paramecium arise from, just now I have explained, this is not basal granules. Just now I have explained. Cilia in paramecium arise from basal granules, pellicles, cytopharynx, trichocyst. What is it? What porifera? Is it porifera? I didn't even teach porifera. So you have to revise. Okay, whatever I am explaining in the class, you have to spend at least 10 to 15 minutes. You have to read the PPT and you have to ask any doubts in the tomorrow's class um, in the class. You have to ask. And so answer for this question. Cilia and paramecium arise from. Very good. Kinetosomes or basal body. Okay. Okay. If you have any doubts, means you can post it now. I'll ask another question. You have to ask me if you have any doubts. Okay. The probable function of contractile vacuole is? Lalita, answer. Is it so? What is the function of contractile vacuole, Lalita? To remove the undigested food particles. What did I say? This contractile vacuole is known for its osmoregulation. What is osmoregulation? Is this mainly doing the osmosis process, which means transporting or transferring the solute or the fluid from the higher concentration to the lower concentration. If the amoeba finds there is an if the contractile vacuole or animal will find many water will be present inside the body, means it has to expel out. That is done by the contractile vacuole. Okay. If the animal finds I need water, which means then the contractile vacuole will suck the water from the surroundings and it takes into the body of the amoeba. Okay. So the answer is to remove the excess water. Okay. Any doubts? Any doubts? Just make it fast. Okay, shall we wind up the class? Just please revise. See, one more thing I have to, once the syllabus is completed, which so not entire syllabus, once each and every topic has been completed in your syllabus, you have to see, for example, in today's class, I have completed the protozoa and its locomotion. You have to write in your notebook protozoa and 5 most important points in the protozoa and general classification. You can refer the PPT for general classification. Okay, let me say, in the locomotion, what is mean by locomotion? First thing, see, you have to read. What are all the locomotory organelles? Name of the locomotory organelles and its types. Examples. Okay, each, for example, pseudopodia, lobopodia, example amoeba, philopodia, example euglypha, reticulopodia, globigerina, axopodia, actinospirium, and actinophores. What is the flagella and what are the examples? What are the types of flagella? What are all, flagella is a locomotory organelle. What is the ciliary movement and what is the structure? What I have explained, you have to remember all those things. And what are the methods of locomotion? Amoeboid movement. What is Sol-Gel theory? Who is explain the Sol-Gel theory? Means Marston Pantin. Flagellar movement. What is the which subphylum subclass will follow this flagellar movement, and what are all the types of the flagellar movement? Example for those types. Like in the same way, you have to read all these things. Any other doubts? What are all the things I have explained in the class? You have to read this. And what I'm trying to say is today I have completed the protozoa, general characters, classification, and locomotion. You have to, your work is you have to take your notebook and write the 5 to 6 important points with respect to the general characters of protozoa. And you have to make a flowchart of locomotion. What are all the first thing, locomotory organelles? What are all the different types of locomotory organelles? You have to make a flowchart or mind map, whatever it is convenient for you. You have to read, you have to take notes, and that notes will give a hand at the last minute. Any other? Okay, any other questions? Shall we wind up the class? If you wind up the class, you can leave now.